Well, we do talk about some pretty cool stuff in this class, but I don't think anything really takes the blue ribbon for cool quite like blood. If we go back to the embryo, maybe 12, 13, 14 days following conception, there is a structure called the yolk sac, and in the walls of the yolk sac, there are hematopoietic stem cells. Those are the stem cells that by mitosis and differentiation can give rise to any of the three types of blood cells that we have, the red blood cells, the white blood cells, and the platelets. Farther along in development, those hematopoietic stem cells will migrate to the liver and also to the spleen and to some degree to the thymus gland. But by the time the baby is born, or at least by 1 year of age, almost all of those stem cells have taken up residence in the bone marrow. So what we have here is a magic mixture of things that can perform a great variety of functions. It is the transporter of oxygen, carbon dioxide. It is the transporter of the nutrients from the intestines, the nutrients such as carbohydrates, fats, cholesterol, water, vitamins, minerals, all the things that we take in by way of the diet are transported to the cells by the blood. Blood is protective. The agents of our immune system, such as the white blood cells and a protein called complement, are in the blood. Blood is also a buffer. It contains bicarbonate ions, which can neutralize the acidity of the extracellular fluid. Blood can also regulate the temperature of different body parts because we maintain a core blood temperature of 38 degrees Celsius. If we need to warm an area up, we run more blood to it. If we need to cool an area down, we clamp off those capillaries and it will cool down because the blood maintains a pretty warm temperature at about 38 degrees. So let's have a close-up look at this magic mixture that we call blood. Remember, it is a connective tissue, so it does have a cellular component. It does have a ground substance, we call that plasma, and it does have a fibrous component, or at least a potential fiber that we call fibrinogen. So let's look at blood up close. Next slide, please. When we take a liquid mixture and we run it through a centrifuge, then those things that are in the mixture will separate out based on their density. So when we do that with blood, those things that are most dense will settle at the bottom. Those things that are least dense will be at the top. So what we see is plasma, the least dense component of blood, should occupy about 55% of the total volume. Down at the bottom, the red blood cells, which are most dense, the number given there, 45%, that's about the mid-range of where it should be. So that's a normal hematocrit or packed cell volume, about 45%. And then in between the plasma and the red blood cells, we have what's called the buffy coat. It's less than 1% of the total. And what we find there are the things of medium density, such as the white blood cells and the platelets. So over here on the right, I have some normal values for blood. The total volume, well, it depends on whether you're talking about a big person or a small person, a male or a female, but the normal range would be between 4 and 6 liters. Less than 4, the person is hypovolemic. More than 6, the person would be hypervolemic. Since volume and pressure are directly proportional to each other, then a person with hypovolemia is going to have low blood pressure. A person with hypervolemia is going to have high blood pressure. The pH of blood, that range is very carefully kept. Remember, the brain has basically no tolerance for a shift in blood pH. So we have a range here of 7.35 to 7.45. So the normal pH of blood is 7.4. Since we are constantly producing acidic waste products, then we have to have ways of neutralizing or buffering uh, the pH, the acidity, so that we maintain a pH right at about 7.4. If it shifts just a little bit to the left or to the right, you're going to faint. If it goes a little too far to the left or the right, you're going to die. So we don't have any tolerance for shifts in blood pH. We maintain the temperature of the blood at about 38 degrees. The hypothalamus is the monitor of that. The osmolarity is a very important thing. Blood has hemoglobin inside the red blood cells. Hemoglobin is osmotically active. It draws water toward itself. Blood also has sodium inside. In the blood is much sodium, and obviously sodium is a water magnet. So blood has a certain osmolarity. When we talk about circulation a little bit later, you'll see how important that is. The blood has to have a pulling force for water in order to reclaim the fluid that leaves the vasculature and goes out to service the cells. Well, that happens at the capillary beds. The fluid leaves the capillary, goes out to service the cells. Well, we have to have a way of getting it back into the circulation, and much of that is due to the osmotic pressure exerted by blood. It is a water magnet, like we would say sodium is. Sodium draws water toward itself. Another principal contributor to the osmolarity of blood is the plasma protein albumin. Since it is a protein, it's made of amino acids, and there are certain people, many people in fact, who have a protein deficient diet. If you have a deficiency of protein in the diet, then you have a deficiency of amino acids. Albumin is made from those amino acids. So if you don't have the amino acids, you don't make the albumin. If you don't make the albumin, then your blood osmolarity is going to drop and your body fluids are going to accumulate in your abdominal cavity. That condition is called kwashiorkor. Also, the viscosity of blood. Blood is syrupy relative to water, so it is about 4 or 5 times as viscous or syrupy than water. Well, you don't want it to be too watery, and you don't want it to be too syrupy. Either way, if it's too thin or it's too thick, it will wear the heart out. So maintaining a normal viscosity, 4 or 5 times as viscous as water, is healthy for the heart. Anything less than that or anything more than that will simply wear the heart out trying to pump it. So once again, albumin, the plasma protein, is a principal contributor to the syrupiness or viscosity of the blood. Well, we have a certain number of red blood cells that is normal. Anything outside of that range is abnormal and therefore unhealthy. So once again, depending on whether we're talking about a male or a female, whether we're talking about a well-trained athlete or a couch potato, so we have a range of red blood cells per microliter, which is normal, that is 4.2 to 6.2 million red blood cells per drop of blood. A drop of blood is effectively a microliter. So anything less than that, well, we're going to call that anemia. If you don't have at least 4.2 red blood cells per microliter, then you're anemic. If you get up around 7 or 8 and there's not a reason for it, now if you are an Olympic athlete and you make more red blood cells because you constantly are physically exerting yourself, well, then it would be normal for you to have a higher red blood cell count than 6.2. But for just an ordinary person, you get up around 7 or 8, the blood is going to become syrupy because of the excess number of red blood cells, and it is high risk for clotting. That condition is called polycythemia. The white blood cell count, 5,000 to 10,000 white blood cells per microliter. The white blood cells are agents of our immune system. So if the bone marrow is not turning out white blood cell counts in this particular range, then you're going to have some problems with your immune system. Uh, Too few white blood cells, you're going to be at risk of infection. Too many white blood cells, usually the problem there, when there are too many, it means that they are abnormal. We're turning out abnormal numbers, and oftentimes the abnormal numbers don't do what they're supposed to do. That is the case with leukemia. We have way too many white blood cells, but that's not really the problem. The ones we're turning out don't do what they're supposed to do. So the person is immunocompromised, even though they have an excessive number of white blood cells. The platelet count given there, 130,000 to 360,000, averaging maybe 250,000 platelets per drop of blood. If you don't have enough platelets, if you don't have enough platelets, you're going to have a difficult time clotting blood. If you have too many platelets, then your blood is going to clot when you don't want it to. So there is a normal range for platelets, and we can't go below or above without getting in trouble. The salinity, that is effectively 1%. 0.9% is effectively 1% salt. That is normal. You can't take just plain water and run it into a person's blood. You'll kill them. There needs to be some salt, roughly 1%. 0.9% is normal saline. So you don't want blood to be too salty. Then again, you don't want it to be too lacking in salt. So there are ranges that have to be very carefully kept. Hemoglobin is our binder and transporter of oxygen. There is a normal range for hemoglobin. We may have a normal red blood cell count. So the red blood cell count is normal, but the hemoglobin value is low. Well, we have to have iron to make hemoglobin. So if the diet is deficient in iron, or if the person is unable to utilize the iron that's in the diet, then the hemoglobin number will fall off, even though the red blood cell count may be normal. So that would be called iron deficiency anemia. We have enough red blood cells, but the red blood cells don't have enough hemoglobin, uh, perhaps due to a deficiency of iron or perhaps due to a deficiency of protein in the diet. Glucose range 70 to 110. If you go below 70, you are hypoglycemic. If you've ever experienced hypoglycemia, you get kind of lightheaded and you feel like you're going to faint. Uh, anything over 110 is hyperglycemia. You are at risk of glucose in the urine and initial stages of diabetes. There is a normal range for cholesterol. Once you get above 220, you're getting into the danger zone for the formation of atherosclerotic plaque. The sodium range, 135 to 145 milliequivalents per liter. Remember that the sodium is one of the contributors to the osmolarity of blood. So we have to have a certain sodium content in order to maintain our blood osmolarity. I listed here the calcium range just to show you how narrow it is. So 9.2 to 10.4 milliequivalents per liter, that is a very narrow range. I mentioned in the hormones PowerPoints the necessity of having blood calcium. If blood calcium begins to fall off, then you're going to go into tetany. If blood calcium happens to be too high, you're likely to form a kidney stone, in addition to having some other nervous system and muscular system problems. So these are just some of the normal values that go with blood. I think a UCA student who has taken human anatomy and physiology should know these. So just stick them under your hat, tell little sister about them, and repeat them until you kind of get it in your head. Well, these are the normal ranges for these particular blood components. Next slide, please. Well, back in the histology chapter, when we talked about connective tissues, I think I made the point many times over that connective tissues all have a ground substance. Well, in the case of blood, the ground substance is liquid. Well, the reason it's liquid is because it's 92% water. So we call this stuff plasma. So what else do we have in the blood plasma besides water? Well, the liver makes proteins. I think it's something like 4 grams per hour that the liver turns out proteins that circulate in the blood. So we call those the plasma proteins. So 92% water, 7% plasma proteins. Some of, by no means not all of, but some of the plasma proteins that we talk much about in this class. Number one, albumin. 60% of the plasma proteins is albumin. It is made by the liver. Since it is a protein, proteins are made from amino acids. Therefore, you're going to have proteins in the diet in order to make this. Remember that the osmolarity and the viscosity of blood is largely determined by the albumin. Also, in the hormone chapter, I talked about transport of hormones in the blood. Albumin is one of those proteins that hormones bind to and receive their transport in the blood. So it's one of the more important of the plasma proteins, and it is the most abundant plasma protein that we have. Another class of plasma proteins representing 36% of the total, those are called globulins. Proteins are called globulins if they are ball shaped. That's all that means. We subclassify the globulins into alpha globulins that would include haptoglobulin. Haptoglobulin will bind to and transport free hemoglobin in the blood. You cannot have free hemoglobin in the blood. If you do have, it will block up the kidney tubules and you will die. So we have to bind it to something to prevent that from happening. So we have this alpha globulin that we call haptoglobulin. Anytime there is any free hemoglobin in the blood, the haptoglobulin will bind it to prevent it from blocking the kidney tubules. Another alpha globulin is ceruloplasmin, that is our transporter of copper in the blood. Another alpha globulin is prothrombin. This is made by, all of these are made by the liver, and prothrombin will come up later in this particular slideshow because it is one of the agents that is necessary for the coagulation of blood. We have another group of globulins that we call the beta globulins. That would include our transporter of iron. The protein is called transferrin. It will bind and transport iron in the blood. Our second beta globulin there, made by the liver, is called complement. I'm not going to go into the details of complement here because when we get to our chapter on immunology, then that's where complement becomes the big player. So for today, it is a plasma protein that is made by the liver, and it is essential to the functioning of the immune system. All right, our third group of plasma proteins there are the gamma globulins. This is the only group that is not made by the liver. Gamma globulins are our antibodies. Antibodies are made by certain lymphocytes called plasma cells. So it's the only plasma protein that is not made by the liver. In this case, it is made by the plasma cells. Those antibodies are made anytime you are exposed to something foreign, and they circulate in your blood plasma. The group of them taken together is simply called your gamma globulins or your antibodies. Our last plasma protein there, constituting 4% of the total, that is our fibrous component or our potential fiber. So the protein is made by the liver. It is called fibrinogen. When we have to clot blood, we're going to convert that plasma protein, fibrinogen, into a net called fibrin so that we don't bleed. All right, so those are the plasma proteins. Then what else is in the plasma besides the water and those proteins? Well, about 1% of what's there. Well, that's the stuff we're transporting. So we're transporting our nutrients. Whatever we ate, we absorb through the intestinal wall into the blood. So those are that's our glucose and our amino acids and our fatty acids and our vitamins and our minerals, things that we eat. Also the electrolytes. Well, we have sodium, we have potassium, we have chloride, we have calcium, we have magnesium. These are charged particles that are in the dissolved in the water of the blood plasma. Well, we produce waste products as a result of our cellular metabolism. In addition to carbon dioxide, we produce nitrogenous waste. That would include urea, uric acid, creatinine, ammonia, creatine. Those are nitrogenous waste products. Well, our waste products have to be transported to the kidneys for elimination from the blood. So also in the 1% there are dissolved gases. We have dissolved oxygen, carbon dioxide, nitrogen. Those are all present in the blood plasma. The hormones that are made by our glands go into the blood, so they are part of that 1% also. And then we have a variety of enzymes that are transported by the blood so that they can work their magic wherever they are needed. Next. Well, good enough. We have plasma. That's about 55% of the volume of the blood. Most of that is water. So blood is liquid. It's about 4 or 5 times as syrupy as water, but it is still a liquid. Floating around or zooming around in the plasma, we have the 3 types of blood cells. The most numerous by far are the erythrocytes, the red blood cells. So these are cells which in their developmental stages kick out their organelles. So these cells do not have a nucleus, they do not have mitochondria, they do not have any organelles. The last organelles to go are the ribosomes, but they also will go. So what we want is a cell that we can fill up with hemoglobin molecules because its principal purpose is to transport oxygen, and hemoglobin is our molecule for doing that. Now, that only works if the hemoglobin is normal. So in the illustration that I have here for you, you can see how the shape of the red blood cell might very well change if the hemoglobin inside is not normal. So this is a sickle cell due to abnormal hemoglobin. Hemoglobin I will talk about on another slide and show you where the abnormality takes place. But because of the abnormal hemoglobin, the cell itself is not shaped right. A normal red blood cell is called a biconcave disc. It is 7.5 nanometers in diameter, and it is about, did I say nanometers? It is about 7.5 micrometers or microns in diameter and about 2 microns thick. It turns out that that shape, that donut type design of the red blood cell is essential for its function. So if we have abnormally shaped red blood cells, chances are they are going to clump up and block the circulation, as is shown here with the sickle-shaped cells blocking the circulation. So if the red blood cells are shaped like stars, or if they are shaped like balls, or if they have anything other than the biconcave disc design, they simply will not circulate. They will clump up and block the circulation. All right, there in the middle, I have the different types of white blood cells. The granulocytes, those that have granules in their cytoplasm, include the neutrophils. Those are the most numerous white blood cells. The basophils and the eosinophils. When we get to our immunology chapter, I will deal with what those guys do in greater detail. Then we have the two agranulocytes. Those are monocytes and lymphocytes. And from them, we get differentiated cells such as the macrophages, the dendritic cells, the B lymphocytes, and the T lymphocytes. So these are the leukocytes or the white blood cells. Now, the white blood cells use the blood as a highway. They leave the blood and go out into the extracellular fluid, into the lymph nodes, into the lymphatic fluid, into the connective tissues. That's where they do their work. So when we talk about a white blood cell count, we're talking about how many white blood cells are actually in a volume of blood. Well, they're just using the blood as the highway. When they get to where they're going, they leave the blood and take up residence out in the mucous membranes or out in the lymph nodes or out in the connective tissues. So we do have white blood cells in the blood, just like you have cars on the highway. But after a while, the cars that are on the highway just end up sitting in a parking lot somewhere. Well, that's the way it is with the white blood cells. Okay, over on the right, you can see this is a microscopic look at blood. The most numerous guys there obviously are the red blood cells. The light shines right through the middle of them because they don't even have a nucleus. Then you have that big blue blob there in the middle. That is one of the white blood cells. That is a lymphocyte. And then the arrows are drawn to the platelets. A platelet is a cell fragment. It is not a cell. It was once called a thrombocyte, but they stopped calling it a thrombocyte because that would mean that it's a cell. Well, they're not really cells, they're fragments of a cell, and we'll talk about what they do later on. Next slide, please. The air that surrounds our good planet is 21% oxygen. We breathe that air into our lungs, and the oxygen diffuses into the blood. Well, the oxygen has to have something to bind to and therefore be transported by to the cells of the body. We have to have oxygen in order to burn glucose. We have to burn glucose because the energy released in that reaction is used to phosphorylate our ADP and form ATP. So the hemoglobin molecule is the transporter of oxygen in the blood. What we have here, shown by what looks like 4 twisted pipes, those are 4 chains of amino acids. We have 2 alpha chains and we have 2 beta chains. The alpha chains contain 141 amino acids. The beta chains have 146 amino acids. Each amino acid chain surrounds what is called the heme moiety. The heme moiety is a nitrogen-containing compound, nitrogen plus several carbohydrate chains, which surrounds a core ferrous ion, Fe+2, that is iron. So each of the heme moieties contains heme, the nitrogenous compound, with the ferrous ion at its core. So it is the ferrous ion which binds the oxygen. So since we have 4 irons per molecule of hemoglobin, then we have a maximum capacity of 4 oxygens per hemoglobin molecule. Well, a typical red blood cell will kick out all of its organelles in its developmental stages. So it loses its nucleus, mitochondria, all the organelles are discharged from the developing red blood cell. The last organelles to go are the ribosomes, so there may be some red blood cells circulating in the blood which still have their ribosomes, but they too will soon be lost. The reason we kick out all of the organelles is to make room for all of these hemoglobin molecules that we pack into the red blood cell. So when all is said and done, we should have about 280 million molecules of hemoglobin per red blood cell. The bone marrow turns out 2.5 million red blood cells per second. That is astounding. 2.5 million red blood cells per second are made, and each cell should contain about 280 million molecules of hemoglobin. Each molecule of hemoglobin can bind 4 oxygens, so that means each red blood cell can transport over 1 billion molecules of oxygen. That is utterly astounding. Well, the amino acid chains make this molecule a protein. Proteins have a certain shape, and oftentimes the function depends upon the shape, which is another way of saying if the amino acid sequence of those alpha and beta chains is not correct, it may alter the shape of the hemoglobin molecule, which may in turn alter the shape of the red blood cell. Well, the red blood cell has a particular function that it has to carry out. If its shape is altered, its function may also be altered. Such is the case with sickle cell anemia. In the case of sickle cell anemia, due to a genetic mutation, amino acid number 6 in the beta chains should be glutamate. But in sickle cell anemia, that one amino acid is valine. So we have only one amino acid out of place in the beta chain, which causes a change in the shape of the hemoglobin molecule, which causes a change in the shape of the red blood cell. So the cells, rather than being biconcave disc, which circulate freely in the blood, the cells take on a sickle type of shape, which have a tendency to get clumped up in the blood, plus they don't bind and transport oxygen very well. So this is how necessary it is for proteins to be synthesized with 100% accuracy. Otherwise, the protein may not function as it should. So when we bind oxygen to our hemoglobin molecule, the hemoglobin transports the oxygen and releases it in areas of the body where the extracellular fluid has become acidic. If the extracellular fluid has become acidic, that means the extracellular fluid is high in carbon dioxide. In areas which are high in carbon dioxide, that means those areas need oxygen. So apparently the hemoglobin has the ability to determine where areas of the body are that need oxygen, and it releases oxygen in those areas. So that is a very interesting thing. How does hemoglobin know where to let go of the oxygens? Apparently, it can read the acidity of the extracellular fluid as it travels through the body, and where there is acidity in the extracellular fluid, that's where the hemoglobin releases the oxygen. Very interesting stuff, I would say. Well, these are cells, and I think we asserted somewhere along the way that there are proteins present in the membrane of cells. Yeah, I thought you said cells were phospholipids and cholesterol. Well, they are, but there's also proteins present in the membrane of cells. Turns out there are more than 100 different types of proteins that might differ from person to person present in the membrane of red blood cells. Well, we have an immune system, and our immune system recognizes what is self and what is not self. So if you take by transfusion someone else's blood and they have proteins present on their red blood cells that are foreign to you, then you might get a transfusion reaction. Now, there are more than 100 of these proteins that have been identified, but there are only a few that cause transfusion reactions. In a transfusion reaction, what you get is called agglutination. The red blood cells will stick together and clump up. Well, if they stick together and clump up, you're going to have your circulatory pathway blocked. That doesn't work. Okay, so let's look at some agglutinogens. The agglutinogen A. Some people have agglutinogen A. That is a protein present on the membrane of their red blood cells. So we say that person is a type A person. They have type A blood. They have the A agglutinogen. Any person who has the A agglutinogen will make anti-B. The proper word for anti-B is agglutinin B. Well, that sounds a whole lot like agglutinogen, so I just called it anti-B, and most people do call it that. So if you have type A blood and somebody tries to put in you type B blood, then your immune system will react against that foreign protein. B is foreign to a person who has type A blood, so you're going to get a transfusion reaction. You cannot take type B blood and put it into a type A person unless you want to kill them. All right, in the second picture, well, we have the B agglutinogen. Some people have the B agglutinogen. That's the protein that's present on their red blood cells. If you took type A blood and you try to put it into that person who is agglutinogen B, the agglutinogen B person makes anti-A. You're going to have a transfusion reaction. You cannot take type A blood and put it into a type B person. In the third picture, we have some people who have both agglutinogens. So they have both the A protein and the B protein. So if we take type A blood and we put it into that person, not a problem. The person says, well, I have an A. That doesn't look foreign to me. If we take type B blood and put it into that person, well, then that person's immune system says, well, I don't see a problem. I have a B. So that's not foreign to me. So the person who is type AB is the universal recipient. You can give them type A blood, not a problem. You can give them type B blood, not a problem. You can give them type O blood, not a problem. So they are the universal recipient. They make neither anti-A nor anti-B. In the fourth picture, we have a red blood cell that doesn't have either the A or B agglutinogen. So it doesn't have either one. If you took type A blood and put it into this person, you're going to get a transfusion reaction. This person's immune system would recognize that A as foreign. It makes anti-A agglutinin. You're going to clump up. If you took type B blood and put it into this person, that person's immune system is going to recognize that B as foreign, and you're going to get a transfusion reaction. So the type O person makes both anti-A and anti-B. Now, since the type O red blood cell doesn't have an agglutinogen, then you can put it in anyone. So the type O person is the universal donor. You can take type O blood and put it in anyone. It doesn't have an agglutinogen to cause an immune system reaction. But you cannot take anything else but O blood and put it into that person. If you put type A, type B, or AB into an O person, you're going to get a transfusion reaction. All right, one of the other proteins that is present, I said there are more than 100 of these, but only a few cause transfusion reactions. This one is called the Rh factor. Some people have the Rh factor, so we say they're Rh positive. Some people don't have the Rh protein, so we say they're Rh negative. Well, if we have a person who has the Rh factor, and you put Rh negative into them, not a problem. There's nothing on the Rh negative cell to cause an immune reaction. But you can't go the other way. You cannot take Rh positive and put it into an Rh negative person. The Rh negative person would recognize the Rh as foreign, and you're going to get a transfusion reaction based upon the Rh factor. There's a whole lot more to be said about this. I would like to talk about erythroblastosis fetalis, or hemolytic disease of the newborn, but I really don't have time, so look it up for yourself. It's actually very interesting, the problems that we can have when mama is Rh negative and baby is Rh positive. Ooh. A typical person has about 5 liters of blood. If that person loses 1 or more liters, then they become hypovolemic. If the volume of blood falls, then the pressure of blood will also fall. If the pressure reaches a point where the tissues are no longer being perfused, particularly the brain, then the person is going to lose consciousness. Well, we have to have ways to minimize the loss of blood. So stopping the bleeding is simply called hemostasis. There are 3 steps in hemostasis which occur in sequence and occur very rapidly. When a blood vessel wall is damaged or broken, then the cells of the endothelium is the inner lining of the blood vessel wall. Those cells release chemicals if they are damaged. Some of those chemicals include serotonin, ADP, and a molecule called thromboxane A2. When those chemicals are released, it prompts the smooth muscle tissue in the vessel wall to constrict. Therefore, it reduces, by constricting or pinching off the vessel, it reduces or minimizes the loss of blood. Point number 2, the formation of a platelet plug. The platelets circulate in the blood, but they never come into contact with the collagen fibers that are in the vessel wall, because the internal lining of the vessel has a coating called prostacyclin, which prevents the platelets from coming into contact with the collagen fibers, the connective tissue of the vessel wall. If the vessel wall is broken and bleeding is occurring, then those collagen fibers are exposed. When the platelets come into contact with those collagen fibers, then there is the release of what is called the von Willebrand factor, which causes the platelets to become sticky and therefore to clump up around the area where the vessel is broken. So that's called formation of a platelet plug. So that would be step number 2. Following that, the processes of coagulation kick in. Coagulation is the actual clotting of blood. The net result of coagulation is the formation of a fibrin net through which red blood cells can't pass. So that is the stopping of the bleeding when we have completed all the steps of coagulation. Coagulation is a very complicated process. There are more than 30 steps, and there are, I don't know how many factors involved, but it is very complex. Don't forget that calcium in the blood is necessary for coagulation to take place. Vitamin K is necessary for coagulation to take place. Well, we have 2 different pathways which come together. If the tissue injury is external to the vessel, then we, by way of the extrinsic pathway, have a way of combining a chemical released by the damaged tissue cells. That is factor 3. It's called tissue thromboplastin. Tissue thromboplastin released by damaged tissues external to the vessel activates factor 7. It's easy for me to remember that 3 plus 7 equals 10, and that's where we're going to, factor 10, the prothrombin activator. By way of the intrinsic pathway, this is inside the vessel in the blood itself. We have activation of factor 12, factor 11, factor 9, factor 8, in the presence of calcium and other platelet-released factors. We come again to factor 10, which is the prothrombin activator. Remember that prothrombin is a plasma protein made by the liver that circulates in the blood. As long as it is inactive, then no coagulation takes place. Well, it is activated by the presence of factor 10. That's why it's called the prothrombin activator. When prothrombin is activated, we form thrombin. Thrombin will convert the plasma protein fibrinogen to an insoluble net called fibrin. So it is like strands of a net which will trap the red blood cells and prevent the red blood cells from exiting from the vessel. So we have formed a clot that stops the bleeding. So those are the steps of coagulation. Now, you are familiar with a disease called hemophilia. So I have on the next slide a little summary of some of the types of hemophilia. If a person has hemophilia, then they are not able to clot blood. Something is missing in the pathways that prevents them from forming the blood clot and therefore stopping the bleeding. So have a look at that now. I think somebody in the class has signed up to do a report on hemophilia, so I don't want to steal that person's thunder. I just want to show you a chart so that you will come to appreciate all of these factors involved in the coagulation of blood. So if we fail to coagulate blood, then we bleed. Well, that condition is called hemophilia, saying that, well, that person loves blood or loves to bleed. Notice that we have two X-linked recessive causes: classical hemophilia, hemophilia A, absence of factor 8, and hemophilia B, absence of factor 9. Well, factor 8 and factor 9 are necessary steps in the formation of factor 10, and factor 10 is necessary for the formation of the prothrombin activator. Well, in these two types of hemophilia, the trait is passed from mother to her sons by way of one of her X chromosomes. So since the male does not have two X chromosomes, the male has an X and a Y, and the Y does not have an allele to outshout the recessive trait. Therefore, the disorder is expressed in males, and it is transmitted by the mother to the son. So hemophilia A and B are both X-linked recessive disorders. In the case of hemophilia C, it is an autosomal recessive trait, so that can be passed on to either daughter or son. Hemophilia C is absence of factor 11. In the case of parahemophilia, absence of factor 5, factor 5 is needed to convert factor 10 into the prothrombin activator. Without the prothrombin activator, we don't form thrombin. So this also is an autosomal recessive, meaning it can be passed to either son or daughter. Von Willebrand disease, absence of the von Willebrand factor, that also is an autosomal dominant trait, dominant rather than recessive. Therefore, son or daughter can inherit the trait. Remember that the von Willebrand factor is necessary for the formation of the platelet plug. So if the von Willebrand factor is absent, then there's going to be problems in coagulation of blood because the platelet plugs cannot form. Once a clot has done its thing and stopped the bleeding and the vessel wall has been repaired, then the clot has to be broken down, and it has to be broken down piece by piece. Otherwise, parts of the clotted blood would get caught up in the circulation. That's called a thromboembolus. When you have clotted blood circulating in the blood, it will eventually reach capillaries that it cannot pass through. Well, that means you just had a stroke, or you just had a heart attack, or you just had a pulmonary embolus. Those things are deadly. So clotted blood has to be broken down, and it has to be broken down in an orderly fashion so that we don't end up forming a thromboembolus that could eventually kill us. So this process is called fibrinolysis, the breaking down of a blood clot and the dissolution of those components of the blood clot so that they don't form a thromboembolus that could get caught in our small blood vessels. Well, the same factor 12 that initiates the process of coagulation in the intrinsic pathway, it's the same chemical that started the coagulation process. Factor 12 activates a thing called plasma kallikrein or prekallikrein, converting the prekallikrein to kallikrein. Kallikrein, in turn, is involved in the conversion of plasmin to plasminogen. There are two other things that I'm familiar with that can do this. The conversion of plasmin to plasminogen can also be accomplished by a chemical called streptokinase. It can also be accomplished by a chemical called tissue plasminogen activator, TPA. So in the event that we have unwanted blood clots, we need a way of dissolving those blood clots before they get stuck in the heart or in the brain or in the lungs and kill the person. Well, kallikrein is our naturally produced chemical for converting plasmin to plasminogen, but as medicinal substances, we use streptokinase, which is produced by streptococcal bacteria, and we also have tissue plasminogen activator. So what we want is to make plasminogen, because plasminogen is what breaks down the fibrin net and therefore dissolves the clot. Remember, we formed the fibrin net when we activated fibrinogen. It became fibrin. The fibrin traps the red blood cells, forming a clot. Well, we need plasminogen to break down the fibrin net piece by piece by piece so that we don't end up with solid particles trying to circulate in the blood. So it is the activation of plasminogen that is the necessary step here. And since we can't always depend on our kallikrein to do that, then we administer streptokinase or we administer tissue plasminogen activator for the purpose of dissolving clots so that we don't have solid particles circulating in the blood.